DNA replication. Replication is the process by which DNA molecules reproduce themselves. They make exact copies or replicas of themselves. And there's going to be a bunch of words here. There's replication, there's transcription, there's translation, and we do need to know what those words refer to. Replication, if you think replica, this is replicating something. DNA replication is making an exact copy of the DNA. So, you think about cells dividing. Each cell needs DNA. And so the DNA has to replicate or duplicate in order for the new cells to form. So the way this happens is that the two strands are going to separate, and each of the strands will serve as a template for the synthesis of a new strand. Remember, these two strands in the DNA are not identical. They're complementary. They have a direction. This is 3' to, this one twists around and comes down here to 5', and this one's 5' to 3'. They're going in opposite directions, and their bases match up. Remember, C and G always pair together, T and A always pair together. So what happens is this separates, and the point at which it separates is called the replication fork. So it kind of unzips almost like a zipper. So I believe it's an enzyme, comes along and opens this up, and then the two individual strands are separated, and the new nucleotides can come in. So DNA helicase unwinds the double helix by breaking the hydrogen bonds, holding the base pairs together. And then those unpaired bases of each strand combine with free nucleotides present in the cell's nucleus, one at a time. So one at a time, new nucleotides come in, and they hydrogen bond with the unpaired bases on the individual strands. DNA polymerase is an enzyme that verifies the correct pairing and catalyzes the formation of a new phosphodiester linkage between the nucleotide and the growing strand. And the result is that you have two daughter DNA molecules. Each of them is identical to the parent, and each daughter contains one strand of the original DNA and one brand new strand. So the parent divides into two strands, and then a complementary strand is formed on each of those strands. So I put a few YouTube videos in the playlist for this class, and so the one that corresponds to what we're talking about right now is the one entitled How DNA Copies Itself. So now we're going to talk about this in a little more detail. So the DNA polymerase, which is what is attaching the nucleotides to each other, only operates in the 5' to 3' direction. So here we have this replication fork. The helicase is unwinding the DNA, and we have what are called, there are two strands. One is the leading strand and one is the lagging strand. The leading strand can grow continuously because DNA polymerase operates in this direction. I was looking at the wrong thing. The daughter strand here is going 5' to 3' because the parent strand is going 3' to 5'. So this one can grow continuously. It can just be replicated as it comes off. The other one is going in the opposite direction, and so that's not going to work. So what happens is some comes off, and it'll start here and go to the end, and then it'll come back and do some more. There is a gap here, which is called nick, a nick in the strand, and that gets connected by DNA ligase, connects the fragments. So the lagging strand grows in segments. It has to form in the 5' to 3' direction, And so it'll make this piece and come over here and come over here. And in the video, that's what it was looping back on itself. So that one has a little bit more trouble forming. The DNA unwinding does not have to begin at an end of the DNA molecule. It often actually happens at several places simultaneously, and the replication proceeds in both directions. And this speeds up the replication of the DNA. DNA is really, really long. What was it? I think it was 3 billion nucleotides in human DNA. So that's a lot of bases. And that video, they were doing that in real time. So even though it seems like it's going really, really fast, if you've got 3 billion base pairs to replicate, that's going to take a while. So what can happen, here's the parent DNA. We can have multiple site replication. So early on, it'll start replicating at several different places at the same time and proceed in both directions, and then it will eventually meet up. And in that way, the DNA can be replicated much faster. So here's a good summary. Step 1, DNA helicase unwinds the 2 strands of DNA. And what happens there is the hydrogen bonds that are holding those base pairs together are broken. Then in step 2, free nucleotides pair with their complementary bases on those template strands by means of hydrogen bonds. So here we have a G base on the DNA, and a nucleotide with C will come and will match up with that because they form 3 hydrogen bonds. They match up very nicely. A and T also match up. They form 2 hydrogen bonds. And so these nucleotides are present in the nucleus of the cell, and they come in and bond to it through hydrogen bonding. Then step 3 is DNA polymerase comes and attaches those nucleotides together. So initially they are just individual nucleotides positioned in place, and then the polymerase comes and joins their backbone together. And then step 4 is the other strand is formed in short segments. I forgot to say this word, but these are called Okazaki fragments, after the guy who discovered them. They're called Okazaki fragments, and those are then joined by DNA ligase. So after the DNA is formed, we've got these 2 new DNA molecules. So we've turned 1 DNA molecule into 2. Then the replicated DNA will interact with specific proteins, they're called histones, to form stable structural units, which are called chromosomes. A chromosome is an individual DNA molecule that's bound to a group of proteins. And it's actually mostly protein. It's typically about 15% by mass DNA and 85% protein. These chromosomes usually occur in matched pairs, one from the father and one from the mother. And so they're called homologous pairs. And each chromosome in that pair code for the same trait. So if you studied genetic characteristics or anything in biology, I only took 1 biology class as a sophomore in high school, but we talked about genetics and the blue eyes, green eyes, brown eyes thing. So in your chromosome, you have 1 chromosome from your father and 1 chromosome from your mother relating to your eye color. And depending on whether one is dominant or recessive, that's going to affect your eye color. Now, humans have 23 pairs of chromosomes. So you had half of them from your father and half of them from your mother. But each chromosome in the pair code for the same trait.